So let's continue from the previous session. So in conclusion, molecular biology and recombinant DNA methods have significantly advanced the field of biology and have numerous applications in medicine, agriculture, and industry. Isolation and purification methods, separation techniques, and molecular cloning are some of the key techniques used in these fields. These methods have helped researchers understand the functions and interactions of RNA, DNA, and proteins, leading to the development of various technologies that have revolutionized the field of biology. Next topic, expression of recombinant proteins and related topics. So, molecular cloning techniques have revolutionized the field of molecular biology and allowed scientists to isolate, manipulate, and study specific DNA sequences, genes, and proteins. Recombinant DNA technology enables the transfer of DNA fragments between different organisms, resulting in the expression of foreign genes in bacteria, animals, and plant cells. So, isolation and purification of DNA, RNA, and proteins. The isolation and purification of RNA, DNA, and proteins is essential for downstream applications such as cloning, sequencing, and expression studies. Different methods are used for the isolation and purification of each molecule depending on the source and type of sample. For example, RNA can be isolated from cells or tissues using phenol chloroform extraction, followed by ethanol precipitation. DNA can be extracted from bacterial or before moving on to that complex topic, let's once again just have a quick recap like isolation and purification of RNA, DNA, and protein. That is the title of this topic. So, the isolation and purification of RNA, DNA, and proteins is essential for downstream applications such as cloning, sequencing, and expression studies. We know that. So, different methods are used for the isolation and purification of each molecule. So, depending on the source and type of sample. For example, RNA can be isolated from cells or tissues using phenol chloroform extraction followed by ethanol precipitation. DNA can be extracted from bacterial or mammalian cells using alkaline lysis, and proteins can be purified from cell lysates using chromatography techniques such as ion exchanges, size exclusion, or affinity chromatography. Next topic, molecular cloning. Molecular cloning is the process of inserting a DNA fragment of interest into a plasmid or the vector, which can then be used to transform bacteria or other host cells for the expression of the recombinant protein. Different methods can be used for cloning, including restriction enzyme, digestion and ligation, PCR cloning, and site-directed mutagenesis. The choice of method depends on the size and complexity of the DNA fragment and the desired expression system. So, expression of recombinant proteins. Recombinant proteins can be expressed in bacterial, animal, or plant cells using different vectors and expression systems. Bacterial expression systems are the most commonly used for the production of recombinant proteins due to their ease of use, rapid growth, and high yield. Animal and plant expression systems are used for the production of more complex proteins that require post-translational modifications, such as glycosylation or phosphorylation. Isolation of specific nucleic acid sequences. Isolation of specific nucleic acid sequences is important for downstream applications such as cloning, sequencing, and gene expression analysis. Different methods can be used for the isolation of specific nucleic acid sequences, including PCR, hybridization-based techniques such as southern and northern blotting, and high-throughput sequencing methods such as RNA-seq and ChIP-seq. Then, generation of genomic and cDNA libraries. Genomic and cDNA libraries can be generated in plasmid, phage, cosmid, BAC, and YAC vectors. Genomic libraries contain large DNA fragments including both coding and non-coding regions of the genome, while cDNA libraries contain only the transcribed regions of the genome. Both types of libraries can be used for gene discovery and functional analysis. In vitro mutagenesis and deletion techniques. In vitro mutagenesis and deletion techniques are used to create specific mutations or deletions in a gene of interest, allowing the study of the function of the gene or protein. Different methods can be used for in vitro mutagenesis and deletion, including site-directed mutagenesis, gene splicing by overlap extension, and polymerase chain reaction PCR-based deletion. Protein and DNA sequencing methods. Protein sequencing methods are used to determine the amino acid sequence of a protein, while DNA sequencing methods are used to determine the nucleotide sequence of a DNA fragment or an entire genome. Different methods can be used for protein and DNA sequencing, including Edman degradation, mass spectrometry, Sanger sequencing, and high throughput sequencing methods. Then genome sequencing strategies. Genome sequencing strategies are used to sequence the entire genome of an organism. So, strategies for genome sequencing. Genome sequencing refers to the process of determining the complete DNA sequence of an organism's genome. There are several strategies for genome sequencing, and each strategy has its own advantages and disadvantages. First, whole genome sequencing, WGS. Whole genome sequencing involves sequencing the entire genome of an organism, including both coding and non-coding regions. This approach has become increasingly popular due to the decrease in the cost of sequencing. WGS is the most comprehensive strategy for genome sequencing and provides information on all the genes present in the genome. It also allows for the detection of structural variations and mutations that may cause genetic disorders. The disadvantages of WGS is that it generates a large amount of data that can be difficult to analyze and interpret. Then exome sequencing. Exome sequencing involves sequencing only the coding regions of the genome. This approach is more cost effective than WGS and focuses on the genes that are most likely to have a functional impact. Exome sequencing is often used for the identification of disease-causing mutations in rare genetic disorders. The disadvantages of exome sequencing is that it does not provide information on non-coding regions of the genome, which may have important regulatory functions. Then transcriptome sequencing. Transcriptome sequencing include sequencing the RNA transcripts of an organism. This approach provides information on gene expression levels and alternative splicing events. Transcriptome sequencing can be used to identify differentially expressed genes in disease states or in response to environmental stimuli. The disadvantages of transcriptome sequencing is that it only provides information on expressed genes and does not provide information on non-coding regions of the genome. Then metagenome sequencing. Metagenome sequencing involves sequencing the DNA of an entire microbial community. This approach is useful for studying complex microbial ecosystems and identifying novel microbial species. So, metagenome sequencing can also be used to study the functional potential of microbial communities. The disadvantages of metagenome sequencing is that it generates a large amount of data that can be difficult to analyze and interpret. In conclusion, the choice of strategy for genome sequencing depends on the research question and the available resources. Each strategy has its own advantages and disadvantages, and researchers must carefully consider these factors when choosing a strategy for genome sequencing. Then, methods for analysis of gene expression at RNA and protein level, large scale expression, and related techniques. So, isolation, separation, and analysis of carbohydrates and lipid molecules are also discussed. Carbohydrates and lipids are essential biomolecules involved in a wide range of biological processes. Various methods for the isolation, separation, and analysis of these molecules are very important. So, isolation and separation of carbohydrates. Carbohydrates are often found in complex mixtures with other molecules such as proteins and lipids. The isolation of carbohydrates involves the removal of other biomolecules through chemical or enzymatic methods. Following isolation, various methods are available for the separation and analysis of carbohydrates. This include chromatographic techniques such as paper chromatography, thin layer chromatography, gas chromatography, and high performance liquid chromatography. Electrophoresis techniques such as gel electrophoresis and capillary electrophoresis can also be used for the separation and analysis of carbohydrates. Then isolation and separation of lipids. Lipids are a diverse group of molecules that include fats, oils, waxes, and sterols. The isolation of lipids involves the extraction of lipids from tissues or cells using organic solvents. Following isolation, various methods are available for separation and analysis of lipids. This include chromatographic techniques such as thin layer chromatography, gas chromatography, and high performance liquid chromatography. Mass spectrometry is also a powerful tool for the analysis of lipids. RFLP or restriction fragment length polymorphism, RAPD, random amplified polymorphic DNA, and AFLP, amplified fragment length polymorphism. Once again, I will be reading this. RFLP, restriction fragment length polymorphism. Restriction fragment length polymorphism. Then RAPD, random amplified polymorphic DNA. Random amplified polymorphic DNA. And AFLP, amplified fragment length polymorphism, are molecular biology techniques used for the analysis of genetic variation within and between populations. RFLP analysis involves the digestion of DNA with restriction enzymes which cut the DNA at specific recognition sites. The resulting fragments are separated by gel electrophoresis and visualized using probes that bind to specific sequences within the fragments. So, differences in the length of the fragments are used to identify genetic variation between individuals or populations. RAPD analysis involves the amplification of random DNA segments using polymerase chain reaction. The resulting fragments are separated by gel electrophoresis and visualized using staining techniques. Differences in the presence or absence of specific DNA fragments are used to identify genetic variation between individuals or populations. So, AFLP analysis is similar to RAPD analysis but involves the digestion of DNA with restriction enzymes before PCR amplification. The resulting fragments are separated by gel electrophoresis and visualized using staining techniques. Differences in the length of the fragments are used to identify genetic variation between individuals or populations. It has some advantages like these techniques are powerful tools for the analysis of genetic variation within and between populations. They can be used to identify genetic markers associated with specific traits or diseases, and they are relatively inexpensive and easy to perform. But some disadvantages are also associated with this, like RFLP analysis is limited by the number of restriction enzymes available and the size of the fragments produced. RAPD analysis can be less reproducible than other techniques. AFLP analysis can be time consuming and requires specialized equipment. In conclusion, isolation, separation, and analysis of carbohydrates and lipids, as well as RFLP, RIPD, and AFLP techniques, are important methods used in biology for the analysis of biomolecules and genetic variation. These techniques have various advantages and disadvantages that should be carefully considered before use. Then, next topic, histochemical and immunology related techniques like antibody generation, ELISA, etc., Western blot like techniques. So, histochemical and immunotechniques are essential tools in the field of biology that enable the detection, localization, and identification of specific molecules in various biological specimens. These techniques have broad applications in fields such as biotechnology, microbiology, biochemistry, and medicine. Then, antibody generation is a crucial step in many immunotechniques, as antibodies are highly specific binding molecules that can be used to detect and isolate specific molecules of interest. Polyclonal antibodies are generated by injecting an animal with a purified antigen, while monoclonal antibodies are produced by fusing antibody producing cells with immortalized cells to generate a stable cell line that secretes a single type of antibody. These antibodies can be used in various techniques such as ELISA, RIA, Western blot, immunoprecipitation, flow cytometry, and immunofluorescence microscopy. Next, enzyme-linked immunosorbent assay, or ELISA, is a common immunodetection technique that uses an enzyme-conjugated antibody to detect the presence of a specific antigen. The enzyme substrate generates a color change that can be measured to determine the amount of antigen present in the sample. ELISA has broad applications in fields such as diagnostics and immunology and biochemistry. Then radioimmunoassay, RIA technique, is a technique that uses a radio-labeled antibody to detect the presence of a specific antigen. The amount of radio-labeled antibody that binds to the antigen can be measured to determine the amount of antigen in the sample. RIA has been widely used in medical diagnosis such as measuring hormone levels in blood. Then Western blotting is a technique that involves the separation of proteins by gel electrophoresis and the subsequent transfer of proteins to a membrane. The membrane is then probed with a specific antibody to detect the presence of the protein of interest. Western blotting is a powerful tool for detecting and characterizing proteins in a sample. Immunoprecipitation is a technique used to isolate a specific protein or complex of proteins from a mixture. The protein of interest is bound to the antibody and subsequently pulled down from the mixture using a bead-bound secondary antibody. This technique is particularly useful for studying protein-protein interactions. Then flow cytometry is a technique that measures the physical and chemical properties of cells and particles as they flow through a laser beam. So, fluorescently labeled antibodies can be used to detect specific cell surface markers, allowing for the analysis of complex cell populations. Then, immunofluorescence microscopy is a technique that uses fluorescently labeled antibodies to detect specific molecules in cells and tissues. The technique involves incubating the sample with the primary antibody specific to the target molecule, followed by a fluorescently labeled secondary antibody that binds to the primary antibody. This results in specific labeling of the target molecule with a fluorescent signal that can be visualized using a fluorescence microscope. In situ hybridization techniques such as fluorescence in situ hybridization, FISH, and genome in situ hybridization, GISH, allow for the detection and localization of specific nucleic acid sequences in cells and tissues. The sequences involve the hybridization of fluorescently labeled DNA or RNA probe to its complementary target sequence, allowing for the visualization of specific genes or chromosomes. Next, biophysical methods, molecular analysis using UV visible fluorescence, Andol, circular dichroism, NMR and ESR spectroscopy, and related techniques. So, biophysical methods are powerful tools used to study biological molecules and their interactions with each other. These molecules are very important, and these methods provide a better understanding of the structure, function, and properties of these biomolecules as well as the dynamics of the interactions between them. Some of the important biophysical methods are discussed below, like UV visible spectroscopy. This method is used to study the electronic transitions in molecules. UV visible spectroscopy can be used to determine the concentration of a particular molecule in a solution, and it can also be used to determine the purity of a sample. This method is widely used in the study of proteins, nucleic acids, and other biomolecules. Next, fluorescence spectroscopy. Fluorescence is the emission of light by a molecule when it absorbs light of a specific wavelength. Fluorescence spectroscopy is used to study the structure, dynamics, and interactions of biomolecules. It can be used to study protein-protein interactions, protein-ligand interactions, and conformational changes in proteins. Then circular dichroism. Circular dichroism is a method used to study the secondary structure of proteins and nucleic acids. It measures the difference in absorption of left and right circularly polarized light by a molecule. CD or circular dichroism spectroscopy is used to determine the secondary structure of proteins such as alpha helices and beta sheets. Then nuclear magnetic resonance or NMR. NMR is a powerful technique used to determine the three-dimensional structure of molecules including proteins and nucleic acid. It is also used to study protein-ligand interactions and protein-protein interactions. NMR can provide information on the dynamics of molecule and the changes in the molecule upon binding to another molecule. Then electron spin resonance, ESR. Electron spin resonance is a method used to study the structure and properties of molecules with unpaired electrons such as free radicals. Molecules with unpaired electrons such as free radicals, ESR can be used to study the kinetics of radical reactions and the mechanism of enzyme catalyzed reactions. Next, X-ray crystallography. X-ray crystallography is used to determine the three-dimensional structure of molecules including proteins and nucleic acids. This method is based on the diffraction of X-rays by a crystal of the molecule. X-ray crystallography is the most widely used method for protein structure determination. Then light scattering. Light scattering is a method used to determine the size and shape of macromolecules in solution. It is based on the fact that the intensity of scattered light is proportional to the size of the molecule. So light scattering is widely used in the study of proteins, nucleic acids, and other biomolecules. Next, mass spectrometry. Mass spectrometry is a method used to determine the mass to charge ratio of molecules. It is used to identify and quantify proteins, peptides, and other biomolecules. Mass spectrometry is also used in the study of protein post-translational modifications. Next, surface plasmon resonance. Surface plasmon resonance is a method used to study the interactions between biomolecules. It is based on the fact that the binding of a molecule to a surface changes the refractive index of the surface. SPR or surface plasmon resonance can be used to study protein-protein interactions, protein-ligand interactions, and antibody-antigen interactions. Then, in conclusion, biophysical methods provide a wealth of information on the structure, function, and properties of biological molecules. Next topic, statistical methods like measures of central tendency and dispersal or related information. So, measures of central tendency and dispersal. Measures of central tendency are used to represent the typical or central value of a dataset. Common measures of central tendency include mean, median, and mode. Measures of dispersal, on the other hand, describe the spread or variability of a dataset. Examples of measures of dispersion include range, variance, and standard deviation. Probability distributions. Probability distributions are mathematical functions that describe the likelihood of obtaining a particular value or range of values for a variable. And binomial distribution is used when we have two outcomes, success or failure in a fixed number of independent trials. Poisson distribution is used to calculate the probability of a given number of events occurring within a fixed interval of time or space. Once again, let's read probability distributions. Probability distributions are mathematical functions that describe the likelihood of obtaining a particular value or range of values for a variable. And binomial distribution is used when we have two outcomes, success or failure, in a fixed number of independent trials. Poisson distribution is used to calculate the probability of a given number of events occurring within a fixed interval of time or space. Normal distribution, also known as Gaussian distribution, is a continuous probability distribution that is commonly used to describe naturally occurring phenomena. Then sampling distribution. Sampling distribution is the distribution of a statistic such as the mean or standard deviation calculated from a sample. It is important because it allows us to make inferences about population based on the sample statistics. Then parametric and non-parametric statistics. Parametric statistics assume that the data are normally distributed and have equal variances. Examples of parametric tests include t-test and ANOVA. And non-parametric statistics do not make any assumptions about the underlying distribution of the data. Examples of non-parametric tests include Wilcoxon-Ransom test and Kruskal-Wallis test. Then confidence interval. Confidence interval is a range of values around a sample statistic that is likely to contain the true population parameter with a certain degree of confidence. For example, a 95% confidence interval means that if we repeated the sampling process many times, 95% of the intervals could obtain the true population parameter. Errors. In statistics, there are two types of errors, like type 1 error and type 2 error. Type 1 error occurs when we reject a true null hypothesis, false positive. Type 2 error occurs when we fail to reject a false null hypothesis, like false negative. Regression and correlation. Regression and correlation are used to measure the relationship between two variables. Regression analysis is used to predict the value of a dependent variable based on the value of one or more independent variables. Correlation analysis is used to measure the strength and direction of the relationship between two variables. Then t-test. T-test is a statistical test used to compare the means of two groups. It is commonly used to test whether there is a significant difference between the means of two populations. Then analysis of variance, or ANOVA. ANOVA is a mathematical statistical test used to compare the means of three or more groups. It is commonly used to test whether there is a significant difference between the means of multiple populations. Chi-square test. Chi-square test is a statistical test used to test the independence of two categorical variables. It is commonly used to test whether there is a significant association between two variables. Then multivariate statistics. Multivariate statistics is a branch of statistics that deals with the analysis of data with multiple variables. Examples of multivariate analysis technique include principal component analysis, or PCA, factor analysis, and cluster analysis. These techniques are commonly used in fields such as biology, ecology, and social sciences to analyze complex data sets. In conclusion, statistical methods are essential in analyzing and interpreting data in biological research. These methods provide a framework for making sound conclusions from data and help researchers to determine whether the results are significant and meaningful. Understanding the various statistical methods and their applications is crucial for designing experiments, interpreting results, and making informed decisions in biological research. Next, radio labeling techniques. So, radio labeling techniques are widely used in biological research to trace the movement of molecules and visualize biological processes. This technique involves the use of radioisotopes that emit radiation, which can be detected using various methods. Radio labeling can be applied to a wide range of biological molecules such as DNA, RNA, proteins, lipids, and carbohydrates. Detection and measurement of radioisotopes. Radioisotopes can be easily detected and measured using various methods such as autoradiography, scintillation counting, and gamma counting. Autoradiography involves exposing a photographic film to the radioactive sample, which results in a pattern of silver grains that can be visualized under a microscope. And scintillation counting involves adding a liquid scintillator to the radioactive sample, which emits light when it interacts with the radiation emitted by the radioisotope. This light can be detected and measured using a photomultiplier tube. And gamma counting involves using a gamma counter to detect gamma rays emitted by the radioisotope. Next topic we will be learning tomorrow. Thank you for listening.